also. And yeah.
Yeah, I just missed it. Like after your talk, after a few months, I've joined the campus. I came from
Okay.
women's college to the main campus.
Gotcha. Gotcha.
Yeah. But I heard your talk when recently last year you've given for the
Oh,
women's
yeah.
college webinar.
Yeah. Yeah. The last time when I came to the campus, 2016, I met with lots of faculty, lots of grant money coming in, and a lot of publications going on, and all kinds of stuff happening.
Mm. Yeah.
Yeah, really nice stuff going on. Let's see if I can get the YouTube link to my classmates. My classmates
Yeah,
wanted to.
I just started, sir. I will share it just in a while.
Yeah, that'll be great. That'll be great. I promised I will send them.
Yeah, I just started the link, sir. I'll share it.
No problem. No problem. Take your time. No worries. I'll be right back. Excuse me. 1 second.
Hello?
Yeah, Padmashri.
Yeah, looks like I got something here. Let's see. Right. Let's see.
Sir, did you get the link, sir? I've just.
Yes. Yeah, I just got it, and I'm forwarding to my classmates.
Okay.
So amazingly, we are still in touch. Some 25 classmates on a WhatsApp group. So
That's
we
great.
chat. Yeah, we chat often, and yeah, YouTube. Yeah, that's right. Okay. Looks like I sent the right one. And this is such a great idea to, bring in mentors and expose them to the current students. And it's particularly good because you see, I need to, because we're all from the same, cut from the same cloth, as we say here. Means that we lived in the same neighborhood and same colleges and high schools and everything. And here we are, we've come this far. So it's kind of a proof, living proof for the current students that they also can succeed. And they also can come this far or whatever, and may even further.
Absolutely,
And,
yeah.
Yeah, even further. And because we started with very little resources, but these days the kids have so much for them available to them. So it's we expect more out of them. So
yeah, they can definitely utilize current resources and go beyond where we are. So
yeah,
that's the hope, and that's what we are trying to help them.
Yes.
Good morning and good evening to everyone.
Oh,
Hi,
good
Mahendra.
morning,
Yeah,
Mahendra.
good morning.
Good morning, Mahendra.
Good morning. Good morning.
Good morning, Mahendra.
Madhvi, Ravi Prakash sir has come.
Um,
Join.
I haven't heard yet.
Good evening, Sarita. Good evening, Leela madam.
Good evening.
Good evening.
Hello, HSP sir.
Yes.
Good evening.
Morning, good evening.
Hi, Sarita.
Good morning, good evening.
Hello, sir.
Yeah, things are better.
Good evening, Bhaskar. Are you there, Bhaskar? Dr. Bhaskar. Good evening, Dr. Arvind.
Good evening, ma'am.
Hello, Dr. Challa Kumar.
Hi, Surya.
Good evening,
morning,
sir.
good
Good evening,
evening.
everyone.
Yeah, your name was referred to
Good
me
morning.
by Shivaguru.
Oh, Shivaguru, yeah, he's a good friend of mine. Sure.
Yes.
Yes.
Good morning, Sudhakar sir.
Good
Yeah.
morning, Amma. How are you?
Fine, sir. Thank you.
Bowling
This is the
Green
time that you
University,
placed.
huh?
Yeah, this is
Bowling
the
Green
right time for
University.
us for the
Hello,
meeting. Actually,
Sudhakar.
I've been telling you.
Yeah. I'm actually giving a talk at Ohio, at Bowling Green this Wednesday afternoon, Sivaguru's place.
When are you doing that? Today there is one by Murthy.
Yeah, today Murthy is giving, and I said I have this other talk, so I will give on Wednesday. So same week,
Okay.
next Wednesday.
Kumar, I have a
Yeah.
question for you.
Yeah, Sudhakar.
There is
Hello.
one. Yeah, there is
Morning.
one string. There's one string. It is being attached to you and me. And I went to the same school, Bazarghat, Nampalli High School.
Really? Wow. When was this?
Which is yours? And I'm a little bit earlier to you.
Okay, mine. Let's see. I graduated I started in 67, and then 68. First year, 68, 69, second year. So I finished in 69.
69 అప్పుడు ప్రిన్సిపల్ శాంసన్ రామకృష్ణయ్య ఉండేనా ప్రిన్సిపల్
తెలీదు మర్చిపోయానండి.
హెడ్ మాస్టర్ మీకు ఫిజిక్స్ సార్ గుర్తున్నాడా?
Marco, so mine is, see, I have to tell you a secret. I will let you in on this. The secret is I was in what is called a special section, 10th Andhra, 11th Andhra. So it was one section that was created for people from current Andhra Pradesh.
Okay. Yeah,
So
నేను
we were not in the-
I passed out from I passed out from there in 68 అన్నమాట 68, so I'm a little bit senior to you.
And 60 past 68 I was 1 year. But
తర్వాత
you must have done 12 years, right? You've done 12 years.
Yeah. No, after you passed out, it is the system has been now discontinued. Multi-purpose
I
has been
know.
discontinued.
Yeah. So you're 2 years older to me because I finished
Probably.
11. Yeah, 2 years old. So
Probably.
I was on the 10th and 11th and so we didn't have science classes, didn't have
Okay.
physics, didn't have anything.
Right.
But another fact is that my sister-in-law became the headmaster of that place
Okay.
years ago.
అక్కడ అక్కడ స్టాండింగ్ ఐకాన్ వాజ్ సుబ్బారావు సార్ ఉండే అక్కడ. I don't know how much you remember. I maintained relation with him for many years. He is no more.
Oh, yeah.
You're talking about 50 years ago, kada?
Oh yeah, of course. Yeah, of course. And then where did you go to for college, Sridhar?
College is మనం ఇద్దరం కలిసి ఇక్కడ టాక్ ఇచ్చినాం కదా, మన ఏది మల్లెపల్లి మన ఓయు ఆ రోజు మీరు ఇచ్చినారు కదా, we were back to back.
MSc
మల్లెపల్లి మన అన్వర్లూమ్ కాలేజ్.
అన్వర్లూమ్ కాలేజ్. ఓకే.
అన్వర్లూమ్ కాలేజ్ లో BSc ఫస్ట్ ఇయర్, తర్వాత సెకండ్ అండ్ థర్డ్ ఇయర్ నేను ఉస్మానియా క్యాంపస్ లో చేసినాను. దట్స్ వేర్ సూర్యప్రకాష్ is my classmate.
That's when we were classmates.
Right.
Yeah. Now I remember everything. Now it makes sense. Aligned quite well. Yeah.
Dr. Vijaykumar, are you in Connecticut?
Yes.
Okay. Not too far from us.
Yeah. Where are you?
I live in Boston.
Oh, of course. Yeah. Hour and 10 minutes. Yeah, sure.
Yep.
Yeah.
Good morning, everyone. This is Ganesh.
Hello,
good morning,
good morning.
everyone. Good evening.
Hi, Professor Ganesh.
Hello.
Hi,
Yeah,
Padmashri. Yeah, I just wanted to say hello to you all again. I'm traveling today. I'm actually my flight is in the evening. So I'll see you in Hyderabad, Padmashri.
Hi, Prasanna. Good morning.
Hello. Yeah, hi.
Hello. Yeah,
Can
we
you
can
hear
hear.
me?
Yeah,
Yeah.
we can hear you. Yeah.
So
Okay.
we'll plan something. Once I come there, I'll reach out to you. But if you can talk to the HOD, Dr. Umesh, please let me know, like any day, like it's good for you all. Like, some people are there in the campus, I can stop by. So one plan is to come on Friday, this week itself. So 7th or 8th also one option that I have to be in Hyderabad and, come to the campus. So if that works out for you, that's a good thing too.
Okay, that's great, Prasanna. If you can go to campus and meet few students, that would be awesome. I will be looking forward for that meet.
Yeah.
Padmashri, can we go ahead and start just to respect everybody's time? We have 62 members joined already.
Yes, Madhuri. Dr. Sarita Reddy is already there here. We can start.
Yeah. Okay. So good morning and good afternoon, everyone. Before I start the event, I would like to wish you all happy Gandhi Jayanti. Thank you all for attending today's 10th mentoring session, which is initiated by OU Chemistry alumni with complete support of OU Chemistry faculty and head of the department. So as we discussed before, I just want to reiterate the purpose of this initiative is to encourage and empower students and scholars to reach out, professors and mentors, and seek guidance in their career and professional development. Well, what that means? What that means is if you guys want to go for a job interview or for any future studies, you need to have good technical background, which is subject, and good resume and cover letter, because those two are the face that you are showing to the interview without showing your face. And also networking and knowing people will be really help a lot. So as a mentor, what we are trying to provide is technical help to answer your questions, or if we are not aware, or if we don't know the answers, we find resources for you to answer the questions and help to prepare the resume and cover letter and prepare for you for an interview, which is how you speak and how you present yourself, your communication skills and soft skills, how you are going to demonstrate when you get an interview offer. So we will help you to build those. And also we try to develop the network to connecting you with the right people so that you will get some help from them to prepare for an interview and how to approach when you are applying for jobs. And apart from these core things, if you need help with any other areas, that we are willing to help you out so that you can improve in your development and also go ahead with your career plans. And we'll try to help as much as we can. So my humble request to all the students and research scholars, please invest some time and take some time and reach out to the professors and also mentors and get the benefit with these resources. So another point I wanted to just put it out there is, in addition to current presentations, what we are doing, presentations with mentors, we also planning to bring some entrepreneurs, because students or scholars might have interest in startup companies, and we are planning to speak some of the entrepreneurs which are part of our mentors and bring them to you so that they can explain their journey when they are when they plan for their startup companies, so that information can benefit you guys as well. And also we are planning to bring some mentees who are part of this program and also other programs, just to give you some information how they grew and how they developed their skills with this guidance and mentoring program. That could be a good direct testimonial, I would say, for you guys to see how it helped some of those mentees who are out there, apart from we as being our own mentees for our mentors. And other than this, if you guys are interested to provide any suggestions or hear any other topics, please feel free to send your suggestions to oumentoringprogram@gmail.com, oucfmentoringprogram@gmail.com, and we'll definitely consider your suggestions. We will take those into give very serious consideration and bring those presentations to you. Email once again, oucfmentoringprogram@gmail.com. We will post this in all the email WhatsApp groups so that you have it handy to you. And before I request Sarita Madam, I wanted to make an announcement. This is a holiday season in India. I know Dasara is one of our favorite festival. So due to the holiday season, we are asking you to enjoy completely. So we're planning to cancel next week, 9th and 16th session, and resume on 23rd with an entrepreneur presentation. So with that, I would like to invite Professor P. Sarita Reddy, Associate Professor and Department of Chemistry, OU and Joint Director of Academic Audit Cell. Sarita Reddy, please.
Thank you, Dr. Madhavi. Am I audible?
Yeah, yes.
Good evening to all, and a very good morning to all the members from US. The Head Department of Chemistry, Professor Umesh. Speaker for today's program, Professor Challa Vijaykumar. Distinguished alumni, dear colleagues, and my dear students. First of all, I would like to thank each one of you for joining this session and extend a warm welcome to the 10th Step Up Mentoring Program on behalf of Department of Chemistry, Osmania University. We have successfully completed 9 programs thanks to our active and dedicated alumni members and Head Department of Chemistry, and also our coordinator, Dr. Hari Padmasri, for their continuous efforts. We are indeed very glad to be associated with our highly qualified alumni members with vast knowledge and experience in different fields of chemistry. This Step Up Mentoring Program, as you all know, it's an excellent platform to know and understand successful and also innovative research, and also the advancements that are going on in the field of chemical sciences all over the world, because we have members abroad and they are sharing their experience and expertise with us. As you all know, science has no bar. It has endless scope and opportunities. I am sure our students will be immensely benefited, and in fact, they'll be motivated as our eminent scientists in the group share their experience and expertise with us. I once again request all our students to participate actively in this program, as this program provides an excellent opportunity to interact with the mentors and then exchange your ideas, then take the suggestions, you get good suggestions about your work, and also establish collaborations. So as Dr. Madhavi mentioned, this is very useful program for your career guidance as well. So before I end, I would like to convey my special thanks to all the alumni members from abroad, because for sacrificing your weekend morning to guide our students. I know how precious it is for you, for all of you, after a long week hard work. Thank you, one and all. I would like to, Dr. Madhavi to take over.
Thank you, Dr. Sarita Reddy. Dr. Padmashri, would you like to
yes,
invite Dr.
Yeah. Good morning, everyone, and good evening to my colleagues here. Good morning to all the mentors of the Alumni Association. And yeah, it's my privilege to welcome Professor Vijaykumar Challa, sir, professor from the Department of Chemistry, Molecular Cell Biology, University of Connecticut, USA. Yeah, he's a proud alumnus of this prestigious Department of Chemistry, OU. Professor Vijaykumar, sir, he started his education from, just now I think he mentioned, from the government high school, Bazarghat. Then he went on to pursue his undergraduation from New Science College, Narayanguda, and of course MSc from the Department of Chemistry, OU. And he obtained his PhD degree from IIT Kanpur under the supervision of Professor M.B. George. He then worked for postdoctoral studies at University of Notre Dame with Professor P.K. Das and University of Columbia with Professors N.J. Turo and J.K. Barton. I think these names are quite familiar. You read about these people, Professor Turo and J.K. Barton, what you have mentioned in your CV, that you have worked with them. And of course, currently is a senior professor at University of Connecticut. He has contributed immensely to the research in protein-based materials to replace synthetic polymers, protein seizers, biosolar cells, biography, and in fact, and its applications it's available on Amazon. It's a very, very informative and interesting book. He also worked on desalination process by forward osmosis, biofuel cells, metal area batteries. And he contributed, he published about 200 articles in peer-reviewed journals, and top 5 papers having an impact factor of 29 nearly. And he gave more than 100 oral presentations, 11 books, mentored more than 20 grants from NSF, DOE, NASA, USDA of USA. And he has several honors to his credit. Won 4 Fulbright Research Excellence Awards, Fulbright Nehru Distinguished Chair Award, Chemical Research Society of India Chemistry Medal, Alumni Research Excellence Award, recognition by Connecticut Governor and Legislature for Nano Art Exhibition. I would like Sir to actually throw some light on this nano art. I am unable to actually share. I thought of sharing those pics he actually sent some time back on the gallery he has exhibited on nano art. That is another way, direction, or no one would think much about that, not much aware of that thing. So I think students would be as well interested. That is the art of science and science of art. It's sir, I would like you to throw some light on that in your talk as well. And he expresses chemistry through his unique nano art, to these exhibits and all. And of course, he enjoys even mentoring school students during summer months, conducting three, and of course, so that means he's very much into this mentoring process. He has already mentored more than 200 undergraduate students. That is a huge number. So to avail his experience even in mentoring, so this is a very, very wonderful opportunity for you, dear students and scholars. We are very much fortunate to have Professor Vijaykumar sir on our mentoring group. So thank you for accepting our invitation. Now I hand over to you. Sir, I will stop my PPT. You may share yours, sir.
Okay, great.
Sir.
Yes. So can you all see my slide? I will go into the play mode.
Yeah, we can see
Yes,
it.
sir.
Yeah.
So can you see the full slide?
Yes, we can see. Yeah.
Okay, great. Lovely. So thank you, everybody. And
one humble request to everyone. One humble request to everyone. Please, who are not speaking, can you please mute your microphones?
yes.
Thank
Good
you so
idea.
much.
Yeah. So thank you, Madhavi, Padmasree, and Sarita Reddy, and so on, and head of the department and OU and all this group of students who have come online, 71 of them, including faculty as well. So thank you all for the invitation to come and speak with you today, this morning, and the evening over there. And the building shown here is our new chemistry building. We call new, even though it's 22 years old. So some names don't change, just like research papers who say novel chemistry, with novel 50 years ago, but still it reads novel chemistry. But it actually looks great, looks brand new. People won't believe it. Anyways, so first is to thank my teacher, my PhD advisor, Professor George. And it's our tradition to acknowledge the Guru Parampara, if you would say. And so thanks to him, I actually could come here and succeed. And he helped me enormously in many, many ways. And in addition to chemistry. So I'm very indebted to him. And just to say that I arrived here with $21, and whatever I have now with me in terms of not just money, but also the professional achievement because of him. And so I'm very grateful to him to mentor me this way. And so I hope to convey some of this back to the group here today. And so most of you are chemists, so I thought my first slide will be on chemistry. But chemistry of proteins, and this you may not have seen. And very often we don't talk about this, and we don't teach the graduate or undergraduate very often. Sometimes we do. So for example, protein is just like a large organic molecule. It is not really different. And except it is very finicky and very sensitive and very, very interesting. And in that sense, we can deal with that as an organic molecule and do chemistry on it. And so, for example, I can attach large chain fatty acids and make a lipoprotein out of it. And these work out to be fantastic nanoparticles. I will talk to you guys how to make this actually in step-by-step manner and what you could do with them after once you made them as nanoparticles. And you could glycate them with carbohydrates or whatever things you want. You make glycoproteins. These are artificial glycoproteins, artificial lipoproteins. And you could make amides out of them, polyamides, for example, like bulletproof material. Polyamides have very strong bonds and then withstand very high stress. So you can cross-link the proteins with a diamine. Standard, very standard chemistry can be applied and to make them. And you can also make nanoparticles much larger, for 40 nanometers and up, very controlled size and uniform, and make them fluorescent and compete with quantum dots. These fluorescent protein nanoparticles are, in fact, in many ways better than quantum dots. And maybe one day might see a TV built out of protein nanoparticles. And you can replace toxic chemicals such as sulfuric acid in batteries being used now with polyelectrolytes made from proteins. For example, acylating the amine groups on the protein can give rise to acids, derivatized proteins. And so they're polyacids and they're polyelectrolytes, and they work really nice as a—
Dr. Kumar, can you make it into the presentation more? So it's only still one slide. So I think your slides are not changed.
Slides are not progressing.
Yeah.
That's hard because I'm seeing different slides.
I think, you know, the window that you selected is the wrong window. So if you go to this on the presentation on left side, top left side, you can see change the screen. So we can only see the first slide.
Sharing is paused for whatever reason. That's the reason.
Can you please reshare it otherwise?
Okay.
Stop it.
Yeah, there you go. Yeah, I saw the button. Oh, I will stop it and then share it again. And let's see, that's the slide. How about now?
Much better, much better.
Yes.
Okay, so now it doesn't show sharing paused. I have no idea. I didn't try to do it.
Now we are at protein chemical modification slides.
Yeah, let's go back then. It doesn't make sense otherwise. But we caught it soon enough. Thank you. So I was saying that the proteins are just like large organic molecules, and you can do standard form chemical transformations on them as long as you're very clear as to what you're doing and control it. And for example, the fatty acids can be attached to make lipoproteins. They're like HDL, LDL, except that these are artificial and they can be used for a variety of purposes, and as single molecule nanoparticles or single molecule micelles and so on and so forth, and as pixels in a display. And you can attach carbohydrate groups to make glycoproteins. Glycoproteins these days are really hot and very interesting for immunology and other applications. So you can make whatever protein you have, you could glycate them with the appropriate chemistry. So again, organic chemistry is the key tool here that we use. And you can make polyamides. You cross-link two protein molecules with a diamine like that, or monoamine, or whatever you wish. And then polyamides are bulletproof materials. And so you can make very, very strong materials out of this as well. And as I was saying, you can cross-link proteins to form very well-defined nanoparticles. And these nanoparticles are potentially competitive with the quantum dots. If you knew quantum dots or familiar with them, you would appreciate how tricky the situation is. Actually, these are better than quantum dots in many ways. And then I will give an example of that if there is enough time. And then, of course, you can make polyelectrolytes by reacting the amine groups with a cyclic anhydride. And then the anhydride opens up to give rise to additional carboxyl groups on the protein. So you have several carboxyl groups on the surface of the protein, making it extremely charged. And you can make, minus 100, minus 150, or minus 200 charge on the protein. These are not naturally available, and these are made only in the lab. And so the chemistry is very powerful. Takes on the natural substance and then converts it into very well-defined systems we want to work with. And you can also wrap them with a synthetic polymer to protect them and improve their stability. We can talk about that. So we early on, we had to solve the stability issue. Most proteins you store at minus 80 degrees centigrade. And so that's not really cool for using them as materials. So early on, we figured out a way to stabilize them so you could boil them, but still they won't denature. So so all that is embedded into this sort of chemical modification approach. We could also make conducting proteins. Proteins generally are not. They are conducting. They are insulators, but because we can modify the substitutions on the surface of the protein or even the interior, we make them conducting. And the other interesting thing, somebody was asking me this question just before the talk, graphene-based modifications. So you can take a insulating protein and then put it in a blender and add graphite and blend it. You get a blend like this: graphene, graphite, and protein. And just 10 minutes and just add water and then blend it in your kitchen. You can do this, and then you can separate the graphite from the graphene, and then graphene contains the protein. And then advantage with that is this graphene is very stable and it's very, very cheap to make. And so we are hoping this will be of some use in the materials chemistry as we proceed. And so, for example, when you take this graphene protein blend and then spread it on a regular printer paper and then allow it to dry, it conducts electrical current, as demonstrated here with an LED. And the conductivity of this sheet, now it's a paper and it is coated with graphene. Conductivity is same as that of silver, essentially. So that means it's a very high value chemical. You start with graphite, which is very, very cheap, and then you convert this into a graphene in a very simple way, and the value of the product increases dramatically. And hopefully someday we can have graphene-based conductors, and they'll be cheaper and stronger than copper-based conductors. And graphene, the protein, I will tell you a little bit about some of the systems we've worked, and then we'll go into one topic and go in depth. So this is kind of an introduction to our chemistry that we're doing. And a while ago, we made a bio-LED with the protein, and then the protein-based bio-LED emits white light as opposed to blue light from a regular LED. White light is a challenge in the LED technology, and so this is a very cool application of the protein chemical modification, which led to this. And more recently, we showed that we can use proteins to make batteries. And so protein metal air batteries, and these outlast lithium-ion batteries by a factor of 2. And also cheaper than lithium-ion batteries. And so sky is the limit in using proteins to make whatever you want to make. And so you can make supercapacitors, a tiny little supercapacitor powering these LEDs for 3 hours after one charge. You can charge this capacitor many, many times. And you can make foams or sponges. And these foams are now being used for desalination of seawater in our labs and testing them, how well they work. And when I come to India, my goal is to commercialize this process, so someone might take it up for desalination in a very, very cheap way compared to current methods. And a plastic kind of a material that we made just weeks ago, protein-based hydrogel that can be converted into plastic. So we're looking for ways to remediate synthetic polymers with protein-based polymers. So this is a protein-based 3D polymer. And so, they already talked to you about this nanoparticles, and they get into the cells and image them, can make all different colors of particles, and the white emitting particles are really special. And we'll talk a little bit about that if there is time. Mostly I will focus my talk on this because a lot of chemistry involved here, very detailed chemistry. So a few slides over there, and then we have done. My group was very successful in making wearable sensors, protein-based wearable sensors. You can practically stitch this into your cloth, and then you can wash it. And in fact, the reviewer wanted us to wash it in a clothes washer 3 times before it is published. And yeah, the sensors still work after you washed it 3 times in a regular washer, commercial washer. And so all this is made possible because of chemical modification of proteins. And then glucose-based sensors, very cheap, very simple, maybe 1 paisa each or something. And proteins and modified proteins and enzymes put onto a piece of paper, and then just like a pH paper, you put the sample on it, and the color develops, and intensity of the color is the proportional concentration of glucose. So this is a glucose biosensor that can be used by people if they have access to. It doesn't need a battery, it doesn't need a meter, it doesn't need a refrigerator. So it gets past many obstacles, and so it can be used in rural areas where there's no power, no battery available. So like this. So a lot of chemistry came out of this, really good chemistry. I'm very pleased with it. Learned so much about the protein-based chemistry. And so we don't do any biology, essentially, and we do all chemistry. Why am I telling you all this? I'm telling you is that if you take a protein molecule and modify in an organic chemistry lab, you can make infinite variants of it, which is not possible in a biochemistry lab. Biochemistry labs, you can make variants, but you vary 21 amino acids, right? So you're limited in your functionality to 21 functional groups that you can, or attachments you can put on. But in the organic chemistry realm, you have infinite number of chemical compounds. You can bring them in and attach them. So you can make so many different kinds of proteins. This opened up a new area for us, a completely new area. So what that means is you can control the properties of the materials at the molecule level by using organic chemistry, biophysical chemistry, and physical chemistry techniques centered around chemistry. So you might ask, why are you doing all this? You know, how did it all happen? So here is a slide that inspired me to do this. And so about 10 years ago, approximately, maybe a little longer, came across pictures like this. You have all this plastic thrown onto the beaches, and and then this was recognized as a problem long ago, but the people didn't pay much attention. And currently there are 5 such. There are 5 islands on the planet, artificially created by humans. Each island is a plastic sea of plastic like that, and it's not a trivial problem because there are about 200 million tons of it. And then every time we use something and throw it away, a part of it gets into the ocean and floats away and then accumulates here. And it's just to show the magnitude, 200 million tons may not mean much to you. We say, oh, well, 200 million, what's the big deal? But it occupies an area larger than South India. So this should spike some interest in you. It means that we have created these artificial islands of plastic. Is causing havoc on nature and environment and many other things. And so we decided we need to do something about it. And so that's when this whole program started out. And so if we can make a plastic or a synthetic polymer out of proteins, then they can degrade very quickly in the environment. And therefore this problem will not exist. And they're green, sustainable, and as I showed you, there's enormous chemical handle on modifying the properties of these things. So how did this happen? Here is a favorite slide I show from a recent review article. So you use the bottles and bags and clothing, they degrade, and we threw them out into the environment. They degrade, become smaller particles, and then it could be microparticles or microfibers. They all get into the waste stream, and then they get into the oceans, and then they get accumulated. So some of this is really important to recycle, but our recycling efforts are the best 12%. And so we are creating a huge problem, which we haven't even started to think about stopping at this point, really, in any concerted manner. And then these things go, if you were to replace this plastic with, say, PLA, that's one of the ideas put forth by these authors, polylactic acid, and the lactic acid polymer, basically. And so it can be decomposed. But in my view, polylactic acid is extremely limited because its properties are limited. You really need to make derivatives of many kinds, and there are inherent issues, limitations with it. So I would advocate replacing this with some other protein-based polymer. And so road runoffs give this, and wastewater, and all this in the end up in the ocean, and we affect the food chain and the fish, and then fish goes into people's diets and so on. And then it also goes up into the air because some of it is burned and some of it is released through other, while you drive on a road, you're releasing nanoparticles made out of rubber, and they're polymer nanoparticles. And now we have detected such microparticles everywhere, all over the globe. And even in snow, when it snows in the North Pole, they have detected polymer nanoparticles being showered down. And so the problem is extensive. So this is a serious issue. And I suggest some of you may be interested in reading this article to get more about this. But today's goal is to go away from this and say, this is our problem. Let's try to find ways to fix this. And how does nature answer this kind of a problem? Nature has been using a variety of materials for millennia, but this problem doesn't exist in nature. That's because very ingenious. Just think about this, just 1 minute. So nature makes leaves. Leaves are biosolar cells, and they take energy from the sun and then store them as food. And we are not able to do this in the lab. We can't construct such a leaf. The leaf degrades very quickly, not an issue. And so if you take the eye of a bird, for example, or an animal, it's a camera, and it is sensitive to different colors and instant recognition. Everything is there in it, and a magnificent device if you were to look at it that way. And birds are flying aircraft, but they decompose in no time. Our planes don't decompose. They are collected in the desert of Arizona, and thousands of them, and so on. And of course, the most magnificent example I can give you about a biological machine is human mind. And this is said to be 100 times faster than our fastest supercomputer. And it consumes about 1 millionth of the power that a supercomputer would consume. And so if we could do good chemistry, this is my challenge to you guys. If you do really good chemistry, we should be able to do this and build computers out of organic molecules or protein molecules. And they would work much faster, much better than silicon-based systems. And it will happen. Maybe it will take a while, 50 years, 100 years, but it should happen. And so where do you get started? You'll start with small problems first and then build in that direction. So for example, I can ask a very simple question. Quantum dots are very popular these days, and everybody wants to make some and use some and so on and so forth. Well, not everyone, but anyways, so the challenge is, well, can I replace that? Most of the quantum dots are made out of toxic elements. And we have not yet seen the effects of that, but it's coming down the pike. We know that. Arsenic, selenium, cadmium, lead, and so on. But that's not a good idea to keep using this sort of thing. And so we can ask, can we use proteins instead to do an equivalent that degrades very quickly in the environment once it is thrown out, but stable when being used. So it should be non-toxic, easy to make, tunable, size, should have reactive function groups so we can do chemistry on them, and so on and so forth. And they should degrade when desired, to be stable under normal operating conditions, and should have optical properties that we can tune them. That's key for quantum dots success. And become nutrients on degradation, not just degrade into ethylene or propylene or butylene, but degrade into amino acids and carbohydrates or nucleic acids that can be used by plants or bugs or whatever, animals, and should be, of course, inexpensive. And so then let's take this challenge, and I will show you one example where we addressed this, in my view, to a large extent. And so let's consider our protein molecule as an organic molecule. Let's assume that we are focusing on the amine groups on this. Also, it has other functional groups, hydroxyls, carboxyls, and other things. So we should be aware of all that chemistry that can happen. And so then if I were to attach a diacid to the amine groups of these things, then I will make an amide group from the amine on the protein with the carboxyl groups on the diacid, and then one free acid will stick out into the solution. The idea here is to modify the protein, but make it extremely soluble in water at the same time. We don't want it to precipitate out for a variety of reasons. By the way, we use water as a solvent in our lab. We don't use any organic solvents, and that's one of the requirements. And in fact, I say that if you can't if you can't make a material that is completely non-toxic, meaning that you can consume it, don't make it in the lab. And so that's the rule. And so we call edible materials because of that. And so another amide can be attached with a second molecule attached to it, and a third molecule, a fourth molecule attached to it. And pretty much you can completely fill the surface with as many molecules as you can fill with diacid molecules. So this now looks like a micelle. Those people who are interested in micellar chemistry would notice this, except that these surfactant molecules are attached covalently. And so this cannot dissociate. It's another dynamic micelle. So coming to the chemistry lab, and if you were to do this, how would you do? Well, we picked a protein, and picking the right protein is important. And this is bovine serum albumin, and serum albumin is a normal protein in blood circulation of mammals, and the most abundant protein, in fact. And this is a waste product from the meat industry, and inexpensive, but it has very elegant structure that I showed you. This is the crystal structure of the molecule. It has various domains here where small molecules are known to bind. And so it carries amino acids and peptides and drugs and many other things. A beautiful carrier, a drug carrier, natural material. And it has, very important to us, a large number of positive charge groups, arginines and lysines. And lysines has a primary amino group on it. So there are 59 of those. And most of them are on the surface, about 40 or so on the surface. So we can modify those primary amines on the surface of the molecule, because once you put it in the water, these amine groups will stick out and they're accessible for reagents. All chemistry is done in water in my lab, as I said. So for example, coumarin is known to bind in this site, and rhodamine is known to bind at this site, and fluorescein is known to bind on the outside of that particular site here. So in other words, most proteins have this weird characteristic, which we don't really understand yet. I mean, they have very specific binding sites for small molecules. They don't bind everywhere. They bind only a particular site. And we have done about 30 years of work in this area. Now we can actually predict which molecule will bind where on a given protein, particularly serum albumin. So the chemistry we do is very, very simple. And we take an acid, RCOOH, and activated with a carbodiamide. And so carbodiamide is a carbon 2 amides on either side attached to it. So imines, bisimine, and then it reacts with the acid to form an unstable intermediate, acylamine, and then the amine opens up with, or is substituted with the primary amine, making an amide bond. And so the acid and the carbodiamide, EDC, is the abbreviation of the water-soluble carbodiamide we use in my lab. So I will use that abbreviation. And so amine, any primary amine can react with it, and then you make an amide bond. And so that's the amide bond. And we use amide chemistry because nature can hydrolyze this. There are enzymes in nature that will hydrolyze the amide bonds. And so we can, whatever we make and throw it away, it will dissociate itself using natural enzymes present in the environment. That's the idea of using amide bonds in all our chemistry. So we start with our protein, bovine serum albumin, so shown in the core here, green. And then you use the EDC I showed you just now, and use this as C8 diacid. This is derived from, let's say, derived from coconut oil. And then you can attach as many as you could possibly, the space allows for it. And so then this is, you have the amide bond, and then the hydrocarbon chain, and then carboxyl group. So same, the chain, carboxyl group, chain, carboxyl group, et cetera, et cetera. So this is an artificial lipoprotein I was talking to you about. So this can be realized in the lab. So now I will show you exact steps, how we have built this step by step. So diacid, so the blue sphere corresponding carboxyl group, and then you have the amines from the protein. And here we have used a N equals to 8. CH2 is 8, and then COOH on one side, COOH on the other side. So C8 diacid. And with the EDC coupling, you can make that artificial micelle, or single protein. We call it a single protein nanoparticle because this has very finite shape, and you can see here, this is a transmission electron micrograph showing that the diameter of the particle is 10 nanometers. And this works out very nicely if you add the diameter of the protein with the twice the length of the diacid, exactly 10 nanometers or so, approximately about 9.5 to 10.5 in that range, based on how you use the conformations. And this is one of the very first TEMs you could say of a organic nanoparticle. That's a very beautiful TEMs come out. And then we can label these with fluorescent dyes and make them fluorescent, just like you have your quantum dots, green, red, blue, whatever color you want, you can make them. They're all fluorescent materials. And so then what we decided, and this was actually done by my student Jingwen Ding. She was head of the department just until a few months ago. Now she is working at Eurofins. So we changed the number of methylenes in our diacid from systematically 2, 6, 8, 10, 12, so on and so forth. And so every time you do that, you're changing the diameter of the particle. And then you measure the diameter of the particle, use light scattering apparatus, and you correlate diameter versus the number of carbons you put in. There's a systematic linear improvement in the size and correlate beautifully, meaning that we're able to make discrete particles that have enough number of carboxyl groups and then long chains on them, so the diameters match up quite nicely. So we can start with about 7 nanometers and it can go all the way to about 20 nanometers. So step by step. And we're increasing the size of the particle using 2 covalent bonds at a time. And that is impossible to, nearly impossible to achieve that kind of resolution, increasing the length by 2 carbon bonds at a time, right? It's very hard and systematically, not by hit and trial, And those who are biochemists may recognize that this protein is chiral, and so it has a circle dichroism. And so before versus after synthesis, you have essentially the same circle dichroism band, means that protein, even though we have taken a hammer and hit on it dramatically, it still retains its structure. This is the beauty of proteins. And this is the size, and this is the gel electrophoresis evidence that we actually changed the size of the protein, charge of the protein, et cetera, et cetera. And so one of the things that we are concerned, as I mentioned before, there is Jingwen. So is that these should be stable. If they're not stable, nobody's going to use them. You can't store it minus 80 degrees centigrade. So what Jingwen did was to take this and then steam sterilize it. Steam sterilization is done by heating at 120 degrees and high pressure. And it kills bacteria, kills viruses, and destroys some biomolecules, most biomolecules maybe, and so on and so forth. It's like a pressure cooker. You just put in a pressure cooker. And what she found is that when you heat it to this, it undergoes denaturation to some degree. But when you cool it, it completely falls right back to its original state. Maybe we do 1% or 2% of the protein. And you can do this all day, heat and cool, heat and cool, heat and cool, many, many, many times. And so what that means is we have changed the biochemistry of this molecule in a dramatic way. Without this chemical modification, if you heat it, it will aggregate and precipitate out. So in other words, it's like making omelet, right, from egg. You throw the egg onto the pan and then it denatures and you get an omelet. The omelet doesn't go back to the egg. But here, the omelet goes back to the egg when you cool it back to room temperature. So this is cool chemistry. And these are some absorption spectra of these different dye molecules. We can make white emitting particles and so on. So that was a 10 nanometer particle. So now you can go back and calculate. If you were to build an iPhone display, what would be the resolution of the screen if each pixel has 10 nanometers in diameter? You will be amazed. Okay, we are not there yet, but one day we will build that. And so then the next student came in and said, well, Bobby Stromer, actually she was before Jingwen, and she wanted to make bigger particles, which is fine. And bigger particles are really good for sensing and many other applications, imaging and so on, because they are bigger, they can have many dye molecules, and so you can give very bright fluorescence, and they can really now compete with quantum dots. And so she made a protein nanoparticle cross-linking, one protein with another, and then labeled them. And if you label them with just right, green, blue, and red dyes, you get white emission. These are the world's very first white emitting nanoparticles of any kind, polymer or protein or whatnot, anything. And you can see these are the actual images of these very beautiful spheres. We didn't expect them to be that way. We thought they will be random sizes and shapes, but they're not. A very sharp size distribution, around 40 nanometers. What is nice is Bobby showed that you can actually control the size from 40 to 170 nanometers as you need. And she's now assistant professor at Wayne State College in Nebraska, and good to her. And she will be taking up this project further, and I say, hey, use this and go with it. And she can take it with her. And here is a gel electrophoresis to show that the white particles are not mixtures of colors of particles. There's a pure white, and then you have blue, green, and red separately. And so this is other evidence. And this is the white emitting characteristic. And those of you who know CIE coordinates, the purity of the white emission is judged by these kind of coordinates. And ideally, you should get 0.33, 0.33 for pure white. But we got 0.33, 0.31 without trying hard. Okay, one afternoon, she made this. And so this is awesome, and we just leave it there. We don't want to go around spending time to make it 0.33, 0.33. And it's very, very hard to get 0.33, 0.33. And people in the field understand this. So this is published in Advanced Materials, Functional Materials. And so what she has done then is to demonstrate that this is a nano pH meter. So because it emits white around pH 7 or something, and you can change the pH, and then the emission changes as a function of pH. And so if you plot in certain intensities versus pH, there is a linear dependence from 4 to 11. And this is the world's first nano pH meter that can detect pH over a range from 4 to 11. Even under normal conditions, we don't have a pH paper that covers 4 to 11. So this is an awesome result. And they get into the cells. And what we asked is that, can we probe the local pH inside the cell at different parts of it? And since we start with white, you see it's a white emission going on in some places. So that is pH is neutral, most likely. Then we see blues and purples. So you can see the blues and purples go this way. So more acidic environments. So there are many, many parts in the cell that are acidic, and then they are neutral, and so on and so forth. So this very clearly illustrates how you could possibly, we're not 100% sure, possibly assign pH to different parts inside the cell. That's the first kind. And it's very simple to do this stuff. And this is green particles, and go into the cells. You can see the nuclei and the interior of the cell very vividly imaged. And so they are being used for imaging. And these are for sale. And so not only that, this is good chemistry, fun, and so on, it's also good in terms of, making some money. And my uncle used to tell me when I was in college that, Vijay, if you made this molecule, we can make 30,000 rupees a kilogram. And if you make this other one, you make 25,000 rupees per kilogram. Now we are selling them for $100 million per kilogram. So my uncle's pledge has been satisfied, and I'm very proud of that. And so you can buy this. You can go to this site, and then they will give you a quote, and then all different colors. You can essentially any color you want, you can buy. Not only for absorbance measurement, but also color in terms of fluorescence measurements. All different sizes from 10 nanometers and up. And so commercial here. So I think I have more or less exceeded my time. So apply to UConn and work with us. And if I can have another 5 minutes, I will show the overview of a few slides. Okay.
Sure, sir. Please go ahead.
Yeah. Okay, great. Thank you. So what we said, and what else can you do with proteins, right? And so, yeah, you can make nanoparticles. You can replace quantum dots. And so here is an idea. So in a summer afternoon, you put a fan on, and you have a bunch of papers, they fly off. So use this idea to make nanosheets. So you have a stack of 2D materials like graphene, molybdenum sulfide, boron, nitride, many, many 2D materials, bulk are available. But if you put a fluid float over them, crystals, you can make nanosheets. This is a concept, and you put a protein in the solution. This is the key player. Then these nanosheets will not come back and aggregate. So you stop the reverse process. And we have used this extensively in the last 10 years. So to make graphite to graphene, as I mentioned to you already, put in a blender, and you blend it for 10 minutes. Sajid is now a senior scientist at Brookhaven National Lab. He's from Kochi and a gold medalist. And he helped me write this book, and he became famous. And we call them Nano Sisters. And Megan is now with Dellinger in Connecticut. And Mansi just accepted a job at ChemX as a senior scientist. What these guys have done is to develop a very simple method, in the lab, just to stir. Take graphite and stir it, or take molybdenum sulfide and stir it, and boron nitride and stir it on a bench top overnight. Next day morning, you have exfoliated protein. Exfoliated materials means nanosheets coated with the protein. Without the protein, nothing happens. So the protein is the key player here, and it stabilizes, and it's all characteristics. You can see nano sheets of graphene here, very cool material. And here we made hybrid of graphene and molybdenum sulfide. That is very highly sought after material in making batteries. And here is the guy, Ankarao, is a current PhD student from Guntur, and he polymerized BSA very first time in the history of mankind to make a polymer film. And so you can see this is a clear plastic. You can see through it, and it's a thin film, and very nice and flexible. And he also made polymer gels, and these are solid, and you cannot compress them. It's very hard to compress these 3D proteins as opposed to 2D synthetic polymers. You can make 3D proteins, very, very strong proteins. It has about 90% water and 10% protein in them. You can make foams. The other thing he did, this is very clever, is that start with multi-wall carbon nanotubes, stir them using Megan's and Mansi's approach, make nano tubes exfoliated in solution. Now add the protein and make the polymer out of it. So now you got a conducting polymer film that has carbon nanotubes, and it's extremely strong, and he used it to make supercapacitors at the same time. So this is our BSA factory, just to show the potential that's going on in the lab. Or you can go one way, see all those, or you can go this other way, see all this. And so in a very nice way, we are able to manipulate the protein, in this case, serum albumin. And this is nothing really spectacular. You can use any protein here. And just to cap off this, is that now Ankarao is using the protein nanoparticles for RNA and DNA delivery to do CRISPR in plants. It's working really well. We put up a patent. That's the reason I couldn't talk about. And another student is using these nanoparticles as carriers for vaccine delivery. She's using mRNA or short peptides and in chickens. So vaccination of chickens is really important, and that's a big market, but it has to be very cheap. And so we are very sure that we could fill that space with it. And so one can ask the question, can we make an iPhone? This is my dream. And the answer is, yeah, partly correct, because we have made a bio-LED, we made a bio-battery, we made metal-free conductors based on proteins, we made protein-based sensors, we made a capacitor. Now we made actually a nice casing as well. And so I think we are getting there in a few more years, hopefully, but you have to make a chip. It's the neuron-based chips. And one of my colleagues at Yale is working on it. And so maybe one day we'll have a completely biological iPhone, which you can, at the end of the day, when it is outdated, throw into soup, make sambar out of it. That's the hope. And so in conclusions, protein-based materials can be simple methods, maybe functional, inexpensive, profitable, can enter cells, non-toxic, sustainable, better than quantum dots in many ways. I didn't tell you guys about that. Biodegradable into amino acids mostly, and so that's very good. New chemistry is coming, really rich chemistry. And a lot of it is organic, a lot of it physical, biophysical chemistry coming out of this. And so these are the people I took pictures on in my last Fulbright and interviewed them. As a little kid, a high school kid, he should be in high school, but he's selling bangles, makes 2,000 rupees a day. And this woman makes 1,200 rupees a day. This guy makes about 700 or 600 rupees a day. And it's not much, but these contribute to the economy of India. 40% of Indian economy is micro these people paid for my science in ways. Same here, we have lots of small businesses to contribute to the science, and the science is funded by these folks. So thank you, these guys. And these are my collaborators, a whole bunch of them. You can't do anything without a collaborator, so you need to have that, and funding from these guys. And these are the people who did the work, and my group. And thanks to Doctors Sri Koti Reddy and Padma Sri and WOI Chemistry and alumni mentors group and all these funding agencies, etc. So also thanks to my wife, amazing on the phone. And without that, you can't do anything. So that's one of the most important things, to have a supporting spouse who stands by you and does all that. So as I was saying a little bit, Padma Sri mentioned, yeah, so we have this art, nano art, and you can see here, these are flowers made out of nanoparticles. Automatically, they just come out that way, and we call hyacinths, or whatever you want to call them. Beautiful flowers. We'll color them physically because they're black and white images, transmission electron micrographs of this. So you can see 10 micrometer is the scale bar on this here. And so these grew out like that. And this is our biohydrogel. And what you see here is actually compartments, and it's 98% or so air, about 2% protein. And so all these are whites, the colors are all different whites. This looks like a nice collage to me. It's like a modern art. And so we can explain nicely how this can be used for desalination of seawater very economically, and treatment. Is iron oxide nanoparticles, and they grow into a tree like that. The very first example of a nanoparticle forming a crystal. It's very hard for nanoparticles to form crystals. So this is a beautiful example. Not only is it rare in chemistry, it's also beautiful art. You can see kind of a fern growing out. And this is a single sheet of graphene. And you show this to elementary school kids, they get really excited. Wow, this is a single sheet graphene, it has 1 atom thick. We can still see it in a microscope. And a couple of other pictures. This is a nice gallery. It was up there for a couple of months in one of these places, and some newspaper put a short note on us. And these are our students, who created the exhibit. I still have some more images here, and this is a beautiful. I have no idea how this is formed. It's calcium phosphate nanoparticles and microparticles put together. They just live it, and they become such beautiful structures. Phase separation, I think, happens in 2 different kinds. So flowers and stems, you can see that. The glucose oxidase is just put as film and then dry it. You get this bullet shot windshield, and it's absolutely lovely. People love it, and we give out the request. People want these images printed out for them. And here is a unhappy Jesus, if you can see. We say that this is actually a lysozyme BSA mixture together. But you can see the nose, and you can see the mouth very angry, and then you have the eyes shut, and then you're wearing a tie, and you can see the tie collar, and then the tie coming down here. And so it's all unexpected. Just nature is the artist here. So like this, and ferns of different kinds and so on. So like this, you could connect with high school kids and primary school kids, and also the parents. Get excited to know what is nanochemistry, what are you doing this, and why is it useful? Because we just wear a white coat and then sit in our lab, do stuff, but people walking on the street have no idea what we're doing and why we're doing. And so it's important we go and talk to them and tell them, look, this is exciting because of this, not just only the artistic value, but also because of the chemistry behind it. And nanochemistry is transforming the world in many, many ways. And so, and that's, I think that's that's the last slide. And so we will stop here, and I can be very happy to take any questions and that you might have. And shall I stop sharing at this point so people can ask questions?
Sure, you can leave this. It looks pretty picture.
Okay.
So, thank you, Professor. I don't know where to start. Just amazed by your work, and
thank you.
It's beautiful, amazing, and I don't think I have enough words to describe what that you guys are doing. like taking an initiative of the utility of the plastic and convert into an amazing art of work, and also in industry and vaccines. So the uses are enormous. That's what I would say. And I'm definitely grateful for your contribution to industry, and also I would say to the globe as a one word, right? So it's amazing work. Students.
Hope so. Hope so.
Yes.
So I hope more people get into the field that then explode it. We want people to come in and explode that into huge area of chemistry.
And I really like the free will of how you want to educate people and let them take ownership and start developing their own thought process and also help others. That's an amazing journey. Thank you so much. I do see, Padmashree, that you have a question. Do you want to read it, or you want students to go ahead and read it?
Actually, the student is a bit shy. Sir, amazing lecture once again. Thank you so much. One of our MSc student, Mr. Fazil, he has actually applied for one small project. He's proposing, for some workshop he's attending in Kerala. So actually, but he's a bit shy person. So I'm asking on his behalf, sir. What he wants to ask is preparing, isolating, and giving a plastic structural support to large sheet of graphene, then embedding it on metal. Can this be done, or has anyone done it already, or is it possible? He wants to make a very large sheet of graphene, not small sheets or small pieces. So he's interested to create a large sheet. So how do we accomplish this?
This is a very good question, and the student is very motivated and very glad that raising a hand and asking a question about this, and wrote also in the chat. I saw the very first question I saw in the chat this morning. And so congratulations to be coming up to this point, and then further on. I'm sure you will succeed very well. So the answer to the question is that it's very hard to make large scale graphene. The graphene that we make about 0.5 micron by 0.5 micron. And that's because the graphite has that size. So we start with graphite, so our graphene is small because of that. And smaller graphenes are a lot of applications to them, a lot of different things you can do. And large 4 centimeters or even 4 inches by 4 inch wafers of graphene are exciting because you could build chips on them, etc. And people are growing those in the industry, and IBM and other places, and they're very expensive, extremely expensive, but it's possible. And in my lab, we use 1 centimeter by 1 centimeter graphene, commercially purchased. It's available. 1 centimeter by 1 centimeter is commercially available. We can buy them, and then the challenge is to transfer, as you mentioned already in your question. How do you transfer it? And so now there are very simple ways to transfer them. And then the industry has taken care of that. So making very large, it depends on how large is very large, right? So if you're interested in making a wafer size graphene, it's going to be very, very expensive. And it's still ongoing. And you do it by using what is called chemical vapor deposition method, where you decompose hydrocarbons, methane or something, and then deposit carbon atoms on a catalyst, and then you transfer that into a substrate that you're interested in. And so that's very well known, and but large scale is still a problem. Large scale
actually, I do have a method. I already submitted to Dr. Padmaji ma'am and Hari Padmashree ma'am.
So that's okay. That's fine, because they don't expect you to come up with a method to make a wafer size thing. It's a workshop, and whatever the apparatuses they have, you have to use them to make what you need to make. And so it's why only single sheet?
I would like to start with a single sheet, and it's not in vapor state. I just want to make it in a plastic structural support. And an alternative method is trying to embed it in a metal without a support of any glue, things like that. And it also has to be thermally stable also.
But the question is why a single sheet?
I have different purposes. It can be made into layers. After I make one sheet, then I could put it on layer by layer.
But that's going backwards. making, as I said, a single sheet is very large graphene, say size of a wafer, for example, is extremely expensive. Why would you go back to make graphite? So what you have to do is this. Yeah, so what you have to do is this. I recommend this. You ask yourself, why am I doing this? Okay, that's the first question. And not just because nobody has done it. That's not a good enough answer. So you have to ask yourself, why am I doing this? And then you come back and say, yeah, is it worth doing? Okay, so only 2 questions you have to ask. If you come up with good answers to those questions, then you go ahead and write your proposal based on that.
There's 2 reasons, sir. The first reason is that it could be made into a wire. Second reason is it could be made as a filter, sir. Graphene, while conducting electricity and without conducting electricity, it has 2 filter capabilities. So I want to experiment with that also.
But you could do all that with regular graphene that you can make from graphite. So you have to justify why you want to make very large graphene. You see what I'm asking question? So very large graphene is very expensive to make. But as I showed you, making 0.5 micron by 0.5 micron is very cheap. But it also does the filtration. It also does conductivity. It also does all kinds of things, wires. You can make wires out of it. You can make an electrical motor out of it. So there has to be a compelling reason why you want to make a very large graphene sheet. And one reason, of course, is to make a faster computer chips. And so if that is what you really want to do, then why graphene? And how can you justify the price? Is it significant to do that? So you have to ask these questions, and based on that, you write your proposal. You don't have to answer all the questions, but you have to think in depth and before writing down.
And also, as per your point, right, Professor, instead of, so when now the cost and also the doability, so can the same scenario can be approached in a different way? Can you make small and make it as like a multiple, so it will do the same purpose? Maybe that, like, because you have a way to do one way, but are there other ways to do the same, accomplish the same outcome? So that probably a good question to pose too. It's just self-awareness and answering all your questions. Yes.
Sorry to interrupt, sir. Actually, what he wants to say is the method now. What you are doing is it's an expensive. You're saying. So he has a method. He has a method by which it would be cheaper to achieve a larger size. He seems to have a method to prepare a larger size and a cheaper method to prepare a large size graphene sheet. That's what he aims at. I think he's looking onto the economics part of it. He wants to make a graphene in a cheaper means. That's what
I get that point. Yeah, I get that point. What I'm saying is that you may have a method to make very large scale graphene very cheap. The way we are making is dead cheap. You can't compete with a method like you stir it on benchtop and you get your graphene. And graphite costs about $2 a pound. So you can make about maybe $5 a pound graphene, single sheet. And so that's the state of the art. So if you want to make something better than that, it'll be very exciting and it'll be awesome. But you have to come up with some logical approach to make it. Right now, the way to make that is using chemical vapor deposition method to make very large size. Means I'm talking about wafer size, or even more than an inch or inch and a half by inch and a half size. So if he has a simple way, cheaper way to do it, that's fantastic. But he has to demonstrate that in his proposal. And then the second question is, if he already has a method to make it that way, what is he going to do next at the workshop, right? So we have to be very clear what is done already in the world and what he has done and what he wants to do, right? So
thank you.
Yeah, so it depends on, it's hard for me to answer it unless I know what size he's talking about and unless he tells me how he made it and why he thinks he can make a very large. There may be a different method that he wants to make. That's okay. So love to hear that.
I think he has to make a statistical comparison of what is actually available right now and what he aims at. Is it really cheaper, and why, as you said, why he wants to go for such a large one? Yeah, that's. Thank you.
And also, like, it's a great thought process. This is how the questions for innovators come. So
exactly.
And definitely the student that speak with the professor, and maybe both of your collaboration, who knows? So
who knows? And yeah, definitely, that's a good idea. And
yeah, actually, I was to suggest him that he collaborates with you. Like, he's having the same. You have the expertise. He has some ideas, and it would be really great.
Yeah, sure.
Exactly.
Yeah. For example, you take these small sheets of graphene, and people have been trying to stitch them together to form a quilt. And people have been trying to do that because there are carboxyl groups at the edges of the sheets. So if you could do edge chemistry, we call edge chemistry on the edge, and stitch them together and make them conducting, then you can make a large sheet out of a smaller sheet, just like making a quilt. And the people are working on it, and partly successful, partly not so successful. And so that's then unlimited. Then you can make any size you are interested in, from smaller. It's like building a wall out of bricks. You just have to figure out how to put a mortar between the bricks and align them. So alignment is an issue, and then doing the chemistry is an issue. So that is doable. If that's what he's thinking, then that is doable. And one has to figure out, how to do that.
Beautiful. There you go. The answer, partially answer, right? So how to link them together. So different ways to do it. There might be other ways to do it. Like, I definitely tell the student to speak with professor and make your way from there.
Yeah.
Yeah.
Try, always try. Yeah. Think and try.
Yeah. Thank you for that clarification, professor, and great question, student. Like, I didn't get your name, but amazing question. Yep. I do have another question here. Is it, Professor, is it possible to make bioelectrodes?
Yeah, I have actually pulled out a scheme where I showed the bioelectrode. It was there in the old slides, and then so what we do is take the graphene and mix it. It already has the bovine serum albumin coated on it, and then we infuse it with enzymes. And then once you're infused with enzymes, you drop cast them onto aluminum sheet or paper or whatever, or carbon cloth, whatever material, or carbon electrode, whatever you're interested in working with. And it forms an enzyme graphene electrode. And this works absolutely beautifully and lovely. This is how we make our zinc protein batteries. And the electrochemistry is awesome, reversible, and fast, and just enzyme electrochemistry is so beautifully shown, like textbook example, using these electrodes. So one of the students graduated with his PhD, is now at, I think, one of the biotech companies in Boston. He just accepted a job last week as a senior scientist. And so he made that enzyme electrodes and did electrochemistry for the first time. I never worked with electrochemistry before. So he's the first one to teach me electrochemistry. And so we did a fantastic electrochemistry project. It was awesome.
Great. Thank you, sir. I see Umesh sir logged on. Just wanted to give him a minute to say hello to everyone.
Sure.
Umesh sir.
Good evening.
Good evening.
Good evening, sir.
My voice is audible?
Yes, sir. Yes, sir.
Yes, sir.
Yeah.
Yes.
So thank you very much, sir. Vijaykumar sir, the talk is very informative, and in spite of your busy schedule, you have given some time to scholars and students of Osmania. So thank you very much, sir. We hope in future also you will give the same cooperation to the department for the development of the department, and you can also suggest some mentors to this group. I hope so. So wonderful, wonderful talk, sir. So we are very happy to see you, sir. Thank you very much, sir. Thank you.
Thank you, Umesh. Yeah, thank you. It's my pleasure.
Thank you, Mr. Yeah. Arvind, I see your hand raised, so I'll let you go with your question before I take another question. Go ahead.
Yes, yes, ma'am. Good morning, sir, and everyone.
Yeah, good morning.
So it's very nice talking. talk,
Yes.
and I have similar my research also is similar to yours, like somewhat
Okay.
different. Like you did white LEDs by using
Yes.
protein, but I did white LEDs by using organic material.
Oh, good. Cool.
So in my PhD, yeah, I'm presently doing postdoc in Wayne State University. That's right. So
yeah. Okay.
My question is, I did what I did for white LEDs to achieve a 0.32 and 0.33 in my PhD. So I used to dissolve my compound in PMMA, and I used to coat it on a blue LED. So for the protein, how you did coated on a blue LED? I want to know that.
Very simple. It's all water solution. So
but it should be dry, right? When
oh yeah,
when
you just dry
coated.
it. Yeah, yeah.
Okay,
You coat it on you dip the LED, take it out, allow it to dry.
Yeah. Okay,
Done.
but what is the stability of that one? Did you measure any efficiency?
Oh yeah, we published this. Yeah, we published a very detailed paper, Advanced Functional Materials, and you can look that up. About I think 2 years ago, and we studied the stability extensively. Not only the stability to air and other things, but also stability to pH and stability to light. And ours was the world record at the time. It lasted 90 hours without burning out. That too present in air, not protected. Most of the white emitting LEDs people make seem to protect them from singlet oxygen damage. We don't have to do that because the oxygen doesn't diffuse through the matrix we put in. The diffusion of oxygen is extremely slow. So we don't have to put any protective layer on it. If you put a protective layer, they will last longer, right? Because we want to have 20,000 hours, not 90 hours for the half-life, 20,000 hours. And you can definitely make it because you protect it from oxygen, and then the degradation pathways are reduced dramatically. And then you look for chemical reactions between the excited states and the matrix that you're putting together. So the sites that are likely to work to quench the excited states or react chemically are disulfide bonds. So what you do, Ben, is to undo the disulfide bonds and oxidize them to sulfonic acids. Then they will no longer be the degradation pathways. So you can systematically shut off each degradation path. And then the tyrosines and tryptophans are the next, there are very few, they're not very many. And so you can modify them chemically as well to shut them off. So you can systematically shut off each photochemical pathway so that you can achieve this much longer half-life. So 90-hour half-life is just like that. And we just didn't do anything just to show the baseline. And we also showed the yield, the lifetime based on white emission. A lot of people don't do that. They take a ratio with the initial blue light, and so their half-lives are distorted because of that. So I'm a photochemist by training, so I know how to do all this stuff correctly. And quantum yield measurements, lifetime measurements, and everything. So I believe you could make much longer than 90-hour half-life. It's not a problem. And it just takes some time and effort to figure out where the energy is leaking out, and also where the excited states are reacting irreversibly to shut off the white emission, right? And so you inhibit those pathways. So it's all chemistry problem, systematic chemistry. You can fix that.
Yeah, and I have one more question. Like you made white emission. You shown one 2017 one paper, Advanced Metal Chemistry, I think. In that you got a white emission from the protein in a cuvette. So how you got that? Like you are mixing a blue, red, green proteins together, or in a single protein you got a white emission?
Yeah, so this is a great question, and a lot of people claim white emission because they have a material that emitting red and then emitting white, blue and then emitting green. They mix them in proper proportion to get white. We don't do that. You know, we said no, we're not going to. That's cheating, because that's not really white emission. That's blue emission, red emission, green emission. So what we were interested in is making a single pixel Okay, single pixel, single nanoparticle that emits all the colors you want. And so we started out with this 40 nanometer particle, protein-based nanoparticle. And then we attached the dye molecules, green, red, and blue, to the same particle. Now, not only just attach one, but we attach about 300 copies of each. And so each particle has about 300 dye molecules of each. So about 900 different dye molecules are attached to one particle. And it's all done in one afternoon. Very good, simple chemistry. Organic chemistry works like a shot here. And then you, of course, purify and get rid of all the others. And so now we have one particle that has all these different dyes. That doesn't mean it should emit white. So what you have to also pay attention to is the excited state properties and the energetics, dynamics of the excited states. Energy transfer happens, and leakage happens, and quenching happens. So many problems are there, and you need to solve each of them one at a time. And then in the end of the day, you can make white emission very successfully. So to answer your question, it's a single particle that emits white. It's not a mixture of 3 different particles. So that is the world's first example to show you can make a white emitting nanoparticles. Quantum dots can't do that. Each quantum dot has 1 wavelength. And if you want 3 wavelengths, you need 3 particles. And so they introduced this solution because they can't make white emission directly. So, but we can make white emission directly. So this is where it trumps the quantum dots. So you have a single pixel emitting white.
Yes. So I have one. It's not a question. Like the same research now you are doing amide chemistry with the protein and organic molecules. Now in my Wayne State also I am doing for mRNA contrast agents. So I'm using WGA protein, and for that I'm making one organic material and binding with the amide process and using for mRNA contrast agents. Yes. It's related, somewhat related to
congratulations.
Your work. Thank you very much.
Congratulations. Wonderful. Good job.
I got some idea also after seeing your presentation. I got some ideas now. So thank you very much for your presentation.
Use it. Use whatever you want. Yeah, absolutely. Go ahead.
Thank you.
Thank you,
job.
sir.
Good luck.
Yeah, and I have a question from student Wani. So her question is, may I know what type of proteins should be selected? Should it have many amine groups in it for linking up, sir?
Yeah, great question. And she's following, obviously, and understanding what's going on, and excited about this. And I just want to take 1 minute and say something slightly outside the talk. And that is, very recently, I started reading ancient Hindu literature. And very recent, Vishnu Purana, for example, or Tripura Rahasya and all that. And I was very fascinated because everything starts with a question from the student. Only then the teacher will tell. Otherwise, the teacher is not going to tell anything. And just wait for the student to ask a question. So this makes me extremely happy that we are following that same principle here. The students have to ask a question first, and then we start answering them. And this is a beautiful way of teaching. And we don't teach that way. And we should teach that way. I really started doing that last couple of years ago. Here in my class, I start with questions. I don't teach unless they ask me a question. So thank you for asking this question, and all the others who have asked the questions. So the answer is that we picked serum albumin from cow because I worked on it for almost 30 years. And so I know very well inside out and all the kind of things. But it also turns out to be one of the best proteins to pick, and because it's a circulatory carrier, so it binds lots of different molecules, and so we can embed all these different dye molecules we want to put in. And it has very specific binding sites in it, and so we could do that. And so about a couple of months ago, we started using other proteins, and so particularly plant proteins. I'm a vegetarian, so I want to do this with a plant protein, just from that perspective. But from environmental perspective, if you can make these devices out of a plant protein, it'll be cheaper, certainly, and more abundant, green, sustainable, etc. And so we started using plant proteins, and you can do the same kind of chemistry. It's not identical as you would expect, because there's different organic molecule. And so if the protein has amine groups, you could do the amine chemistry. If the protein has carboxyl groups, you could do carboxyl chemistry. You don't necessarily have to have only amine groups. And if you have hydroxyl groups, you could do hydroxyl chemistry. So whatever protein you pick, you ask, what functional groups do we have on its surface, and how many of each kind? And then you ask, which ones are accessible for chemistry? Some are not reactive for reasons we don't know yet, but most are reactive. And these days you could use a program called AutoDoc to test out if some chemical modification, you can actually do it. So you do this in silico first, and then you get a really good result saying, yeah, I can put up to 15 of these around my protein, then you're ready to go and then do it. And these days, the computer algorithms are so good, and within a few days, you get the answers. And so I would suggest, if you have access to a supercomputing facility, you do that first. You learn so much from that, and then you translate that into the lab chemistry, and then you walk into the lab and then do the experiment. It is likely to work. So you can use essentially any protein, but I would say, use proteins that are water soluble, because we don't want to get into the problem of using organic solvents and then trying to recycle or deal with the organic waste that come out of it. And in my lab, we have taken a pledge that we don't use organic solvents. We also pledge that we make anything almost edible only. We will not make anything that is toxic and harmful. I think those are good moral rules, not science, to follow, but it's up to you. So take any protein you want, and if it is soluble in water, I would say do that. It's much better than using proteins that are lipid soluble, membrane soluble proteins. There are many of them. It doesn't mean that you cannot do that. Yes, you could, and you should if necessary. So, yeah.
I like your pledge. It's really,
yeah.
it's really so these are some of the very simple concepts, but that take you a long way for your values and morality towards
because we messed up this planet so much. You have to make some drastic changes.
Exactly. Thank you, sir. And I see Mahinder, your hand raised. Please go with your question.
Thank you, Madhavi. Again, always the students are the first priority. We can ask questions anytime. So please, don't worry to, don't think otherwise. Just go ahead and prioritize students' questions first. Madhavi, I just saw your message.
Thank you.
Ma'am, can I, ma'am? I'm Vani here.
Go ahead.
Yeah, Vani, please go ahead. Yeah.
Sir, I'm a research scholar and working under Dr. Vasavi, ma'am.
Hi, Vani.
As you said, it was very informative, sir. Very we will be doing on it. I'm working under molecular modeling lab only, sir. You do all the Autodesk and all. Sir, as you said, there are some active sites to bind up. Is there any specific active site every time for a protein to do all these, to build up some biomaterial or to make some biomaterial? And any specific active site, or only it depends on proteins, whether I'm not getting it. Is it protein specific active site or only common active site is present?
Yeah, okay. It's a great question. Is it specific to a given protein, or can we do this in different ways? And the answer is what you want is define your problem first, right? So for example, I want to make a protein that is conducting and conduct electrons, for example. Then I need to attach certain functional groups that will enhance the electron mobility in the protein. And so I can go back and say, chemistry-wise, what molecular segments increase the conductivity of the material? And this is known already. And so I can bring those and attach them. Now I will ask, which protein should I pick? Well, it depends on what functional groups I have on my molecule I want to work with. So if the molecule I work with has say carboxyls or amines, and it's fantastic. It's easy to work with. But what if it doesn't? If it has only, say, unsaturated double bonds and triple bonds, then you need to introduce some chemistry into your protein in order to connect that. So you could do a 2+2 addition, for example, or you could do 2+4 cycloaddition, photochemical or thermal. And so you need to design a chemistry that works with it. So whatever protein you have, you begin with it. But the protein you want to pick should be inexpensive and should not be toxic and should be green, sustainable. And in terms of industrial production, right? One day you want this, everybody to use this. Then you want to make it in millions of tons. And right now we make 300 million tons of synthetic polymer. So you want to compete with that, means that your industrial base has to be that large. And there are a lot of plant proteins that can meet that. Animal proteins, unfortunately, don't have that kind of industrial base. So that's another reason to switch to plant proteins. So I would, I'm biased, and I would suggest using plant proteins. And then any protein will do, as long as you understand the surface chemistry of the protein. I mean, a lot of proteins, the surface chemistry is not well understood. So this is the problem. And then your difficulty will be to solubilize it in water. And many of them soluble highly, and so you can use them, but not all. And so, as I said, you can do anhydride chemistry to introduce carboxyl groups on the surface. And this works fairly universal in many, many, many proteins. People have done this. Succinylation works. And succinic anhydride is biocompatible. Succinic acid is used in biology. And so even if you release succinic acid subsequently, it's good, it's okay, not harmful. So you can convert a partially soluble or low solubility protein into high soluble. Now you created this active site that you're interested in, that you're talking about. So if the protein doesn't have an active site, you can make it through chemistry. So as I said, it's just infinite possibilities, right? So you can do anything you want with this kind of latitude in chemistry that we have. Biology doesn't have that. And so this is where we champion over biology. Chemistry wins over biology here.
Thank you, sir. Thank you so much. And like I saw one of your slides that your protein that you have taken is about lysine, or about 28 or 30 lysines lysine groups are there. So lysine is very important in a protein to act to make up a biomaterial, or just like that.
So you could look in the database. They will show you how frequently lysine appears in a protein, how many, and it's not necessarily you have lysine. You can have aspartates and glutamates, and they also do the same chemistry but backwards, right? So instead of attaching an acid, you attach an amine to it. And so you can modify the carboxyls into primary amines. We have done this as well. You reacted with ethylene diamine first. So they convert carboxyls into primary amines, and then you attach the things to. So it's like, you may have heard this story. That is, I think this is really good lesson I learned by listening to this news story. Some kid in West Bengal or something was preparing for his 10th class exam, and he was asked to write an essay. And he prepared just one essay on a cow. He didn't know anything else. But when he went to the exam, that question didn't occur. The question was, write an essay on moon landing. And so the guy didn't know anything about moon landing. And so, but he had a presence of mind, and he said, so man landed on the moon and then saw a cow, and cow has 4 legs and a tail and gives milk. And so he wrote his entire essay on the cow. But the opening sentence was fantastic. So this is what I tell, do this in chemistry. So if you know amine chemistry, do amine chemistry. If you know carboxyl chemistry, do carboxyl chemistry. So take whatever problem you have and then approach with it from your point of view, and you will be phenomenally successful. And so you got to understand what you have, what you can do, and apply that straight, your strength into the problem. So if you don't have lysine, no worry, use the carboxyls. If you don't have carboxyls, use hydroxyls, no worry. So you manipulate your problem in a way it will be accessible to your strategy. So adaptation, science adaptation.
Thank you, sir. Thank you so much. Thank you so much.
Thank you, sir. Mahendra, go ahead.
Yeah, go ahead, Parveen.
Yeah. Good evening, sir. My name is MD Parveen, working as a PhD scholar from Osmania University, sir, under the guidance of Dr. Alia Madam. Actually, my question is, sir, here, You told in the presentation when the micelles we are taking as a big micelles. Okay, in that you are going to add the peptide bond, but we are taking is the 2, 4 like even numbering of that carboxylic acids.
Yeah.
Why can't we add the odd number of carboxylic acids? I got a doubt, sir, there.
Yeah, so no worries. That's a great question, and you can add odd, even anything you want. We just picked because they're commercially available. My student could buy off from Aldrich. So 2 carbon, 4 carbon, 6 carbon, 8 carbon diacids. And in fact, first we started with monoacids, and that works beautifully as well. But diacids are water soluble and easy to work with. And so we were able to put in from C2 to almost to C18 or something. And you can buy even longer chains if you're interested in. So it can be odd or even, it doesn't matter. Whatever, we didn't care, right? We wanted just to show that you can increase the length systematically of the surfactant or the acid, and thereby increase the size of the particle systematically. Tuning the chemistry, right? Tuning the size go together hand in hand. So you could use amines instead of acids. Diamines will work the same way as diacids, or dianohydrides will work the same way. So you can extrapolate this chemistry to many, many scenarios. So there's nothing specific. It's a very general concept, and it should work with different things. Unfortunately, we don't have time to do everything, right? We want people like you and others to go in and say, hey, let me do this, let me do odd. So fine, go ahead. And we need a lot of people to develop this chemistry. Remember, we want to build a brain in the lab chemically. So we need a lot of chemistry help, a lot of chemistry we need to learn.
Okay. Thank you, sir.
You're welcome.
Thank you, sir. I think due to time constraint, I have to hand it over to Dr. Padmashri. Padmashri, do you want to take it over from here?
Yeah. Thank you, Madhvi. Thank you very much, sir. Can you please stop sharing your screen? I just
sure. There we go. Stop sharing. Did it work?
Yes.
Yes.
Yeah.
You can share it now. Yeah. Thank you.
Is my PPT visible?
Yeah.
Yes, we can see. Yes.
Yeah, thank you very much, sir. This is a small appreciation certificate from the Department of Chemistry. So I just post it to your email. May I request Mr. Manoj Kumar to present a brief vote of thanks, formal vote of thanks to sir.
Okay, is this audible, ma'am?
Yeah, please carry on.
Myself, Pallati Manoj Kumar, is a scholar under supervision of Professor T. Pathsarin, sir.
Hi, Manoj.
Hi, sir. On behalf of research scholars and students, good evening to one and all gathered here on this wonderful evening and morning. I would like to present the vote of thanks. We take this golden opportunity to thank the respected chief guest of this alumni session, Dr. Challa Vijaykumar, sir, for attending this alumni association in his busy schedule and sharing personal and professional experiences. Sir, giving excellent information, protein chemical modification, biological materials, and single protein nanoparticles. We will cultivate the advice you gave us in real life. Thank you, sir. I extend my gratitude to the respected head department of chemistry, Professor Umesh Kumar, sir, Osmania University, for conducting such a informative lectures and interaction sessions. It's my pleasure to thank Dr. A. Haripadmasri ma'am for her motivation towards the interaction with such eminent mentors who are from Osmania University. And also I thank to mentors and alumni association members and research scholars. Once again, I thank you one and all present here. Thank you.
Thank you.
Say thanks to co-hosts Madhavi and others.
Oh, sure, no problem.
Sorry.
Yeah. Don't worry, Manoj. It's
because they are the main pillars.
Yeah.
Pillars.
Sorry, sir.
Without them, nothing runs.
Program. Okay, all co-hosts, particularly Madhavi madam.
No problem.
Thanks to
it's our pleasure to be bringing presenters and students participating, that is like a more pleasure for us. So appreciate everybody's effort. Yes, it's a team effort, I would say. It's team effort.
Yes.
Yeah. Yes.
Wonderful. Great job. You guys are doing an amazing job. Absolutely lovely. I wish my department did this. So I was telling my students, we should have done this long ago. We have all the resources, but because we need people like you to take the time and initiative to do it. Yeah.
Thank you. Thank you. It's everybody's effort. It's absolutely team effort. Yeah. Thank you. So I would encourage all the mentors, due to time constraint, we couldn't go over your questions, but please feel free to reach out.
No worries. No worries, Martin. No worries.
No worries at all. No worries at all. In fact, I was not even asking a question. I was actually making a comment. About his guide, Professor M.B. George.
Yes.
If you are not known Professor M.B. George, you really don't know Professor M.B. George. He is one person. If you don't know him, actually you don't know him. Because but I'm sure, Dr. Challa Kumar learned this meticulous planning and execution from his guide. He's still there. He's there in Thiruvananthapuram. A grand old man, still active. He's single, not married, and he comes to the lab every day. So, although he is not coming every day nowadays, but I listened stories about. I met him several times, but I did not work in his lab. They're all true. If you survive with him, I think you can survive anywhere in the world.
Anything, absolutely. I mean, this is absolutely rigorous military training.
Even if he's a speck on the table, the student's life is gone on that day.
No, I remember you. I'm glad you mentioned this. When somebody wrote methane on the board using a chalk in those days when I was in graduate school in the 80s, late 70s. You write methane, CN4 hydrogens on 4 sides of the carbon. All the hydrogens have to be identical in size.
Yes.
If one hydrogen is not, you will ask, is that hydrogen different from rest of the hydrogens? So that is the extent of detail he wanted us to think about, and then do it. And he would shout like hell if you don't follow through. And it's just absolute.
Really, I don't think there is anybody like Professor George in this world. He was so meticulous. Even when he comes to IISc Bangalore, where I saw him so many times, people have fear. He's a giant of a person. He's very highly built, well built.
Yeah. And, if you think he is great, think about his mentor. His mentor is Rolf Huizinga, the Huizinga reaction. Rolf Huizinga. So George wrote a review article and sent it to him to read. And it was in the 80s. So that guy is even more meticulous than this guy. He cut the thing into pieces and then he pasted them and sent it back here. Sentence by sentence, he cut it and then edited it that way and then sent it back to him. So he would get it retyped in IIT. So it's just awesome, it's Guru Parampara. That's what I keep saying. It's awesome Guru Parampara. It's indebted to my teachers. Many things I've learned from them.
Absolutely. I don't want to take more time. I'm sure people would like to leave. But amazing talk and very good, meticulously planned, lucidly given, more importantly. Absolutely lucid.
Thank you.
Thank you, sir. Actually, Padma Shri, I wanted to ask you a question. Is this connection is going to stay for a little longer or is it going to time out?
No, there's no time out. It will continue as long as we continue it.
Okay, so then I think if whoever wants to stay here and have a conversation, we can have that open.
Absolutely. I'd be happy to stay back.
Okay, so
I have to go because Dr. Vijaykumar knows this. At 8 o'clock, we have another
I would suggest, Madhvi, the mentors can have this conversation on the WhatsApp chat. Let me please end this session now for today.
Okay, sure. That's fine. Sure. Yeah, totally.
Whatever works.
Thank you very much, sir.
Thank you, everyone, and have a great weekend and great evening and afternoon, whole day. Thank you very much. Bye.
Thank you.